All right, I'm going to share my screen and do you see in general?
Yep.
Yes or no?
Yes.
Questions, coronavirus and so on? Okay, good. Then we're all on the same page. I have the questions ranged across a wide array of topics, some of which were directly on the topics that I had designated, others were more general. So some of the general ones are really fundamental to the course, and so I'd like to go over them since there does seem to be some uncertainty or confusion. So I'm going to go over those first. And the first one here is, what is the difference between the ribosome and the rest of the RNA? So in responding to this, I refer you to this slide taken from the lecture. Actually, two slides here. One shows the three different types of RNA that are involved in protein synthesis. The messenger RNA, shown here as a little strip. The ribosomal RNA, shown here as two subunits, large and small. And the transfer RNA, which is the one that transfers each amino acid to the growing peptide chain. There are 20 of these transfer RNAs, one corresponding to each of the 20 amino acids found in protein. So how does this work? What is the function of each of these RNA types? Well, if you go down to the next one and look in the upper left corner, you see the components of the machinery required for protein synthesis. You have the transfer RNA with a specific amino acid attached. You have the large and small subunits of ribosomal RNA, and you have the messenger RNA, which specifies the sequence of a particular protein. If you move to the right, you see them all come together. The large and small subunits of ribosomal RNA are like a little machine that ratchets itself along the messenger RNA, reading one codon at a time. And at each codon, it corresponds to the anticodon on a transfer RNA. So when they bond, the first amino acid is specified. And then as it moves along to the next codon, the second amino acid, and the third and the fourth, and finally you have a chain of amino acids as the ribosome moves along the mRNA, reading the code. After when the ribosome comes to a stop codon, it stops and releases this peptide chain which it's made. That goes off to do whatever job it's supposed to do, whereas the components of the synthetic machinery get recycled. They go back and start all over again. So I hope that makes it clear what the different types of RNA are that are involved in protein synthesis, and how they work together to make a polypeptide. Any question about that? All right, another basic question that came up a couple of times In one case, the question was, what are the 6 steps of translation? And I think what the questioner was really asking is, what are the 6 steps of DNA replication? Do you recall the video? And I encourage you to go back and re-watch that video because it makes it very clear how the procedure of DNA replication goes ahead. There are 6 components to the factors required for DNA replication. They're listed here. So this picture I'm showing is a screenshot from that video. You have the DNA that's going to be replicated, and an enzyme called helicase. Its function is to separate the DNA strands, because DNA polymerase cannot get in there to replicate as long as the strands are hydrogen bonded to each other. So helicase separates them. Then to keep them separated, there are the SSBs, single-stranded binding proteins, which bind to the DNA temporarily, just long enough to keep it separated until the synthesis is done. And there's an enzyme called primase, which makes a primer. And in this picture, the primer is this little red segment right here. It's a very short piece of RNA, not DNA, short piece of RNA, which must be on both the leading and the lagging strands. Then the workhorse that does the job is DNA polymerase 3. Now it can't do its job until the first 3 do theirs. And this works just fine on the so-called leading strand. Remember, DNA polymerase can only go from 3' to 5'. So with this primer here, it knows, okay, that's where it has to start. And it goes on laying down nucleotides, new nucleotides corresponding to the template. And as it moves, the helicase is moving ahead of it, opening up more and more sequence to be copied. And that process continues all the way from the 3' to the 5' end of the template strand. And that's all that's needed. The problem arises because of the requirement that DNA polymerase has of going 3' to 5'. So on the other strand, the so-called lagging strand, it's now backward. So it can't just start at the 3' end because that end hasn't unwound yet. Helicase hasn't got there yet. So while the leading strand is getting replicated in one continuous motion, the lagging strand is doing it in steps. So as the helicase opens up the double-stranded DNA, so it's single-stranded, primase lays down a series of primers, a short distance from each other. And once the primer is there, DNA polymerase comes along, and here's where RNase H comes into play. It has to remove the primer so the DNA polymerase can go ahead. So the DNA polymerase makes a short segment of DNA, of new DNA, called an Okazaki fragment. There's always a gap between the Okazaki fragments, and DNA ligase fills that gap. It's just a gap between adjoining base pairs. So that's the point I want to make about the leading and lagging strands. One of them, DNA polymerase, is continuous. The other, it's discontinuous because it's going backward. It can't replicate DNA that's still hydrogen bonded. And this next picture here is just a close-up, a very simplified version of that happening. The leading strand going 3' to 5', continuous, and the lagging strand being disjointed, this series of primers, series of Okazaki fragments linked together with ligase. So is there any uncertainty or unclarity about how DNA is replicated? The bare bones of it. Speak up if you have any questions. If not, then I will go to the next question. There were a number of questions, all dealing with types of mutations and how do you recognize which type of mutation you're looking at. Well, for that, I refer you to this slide taken from a lecture, which has the different kinds of mutation listed and says what happens in each of them. Now, to help you visualize this, I've illustrated each type compared to wild type. So this is the wild type sequence that I took from the recent quiz that you had. And I've got 4 different types of mutation, each of which I've illustrated. So the wild type sequence is on top. If you go to the next one down, it's perfectly matched up with the wild type sequence until you come to this GGA. So there's an ACT in the wild type, ACT in the mutant. There's a CAT in wild type, CAT in the mutant, and the rest of the codons are all the same. The only thing different is this GGA is missing. It's been deleted, and so this is called a deletion mutation. The next one down is a duplication. Again, the sequence of a duplication is identical to wild type as you go along the chain, and then at some point, this GGA is there, but instead of the next codon being a CAT, it's another GGA. So the GGA codon is duplicated. After the duplication, the sequence goes back to wild type again. CAT, CGT to the end. Now, I'm showing all these as 3 base pairs. That's not usually what happens. I did that for simplicity, but a duplication, for example, can be much larger, it can be kilobases of DNA, as can an inversion or an insertion, or a deletion. Deletions can be very large. So how do you spot an inversion? Well, you look at the sequence of a mutant, you compare it to the wild type sequence, and you'll see that it's all the same down to this ACT, same as wild type. But then instead of GGA, you have AGG. It's backward because the piece of DNA inverted, it flipped over. After that inverted codon, it goes back to wild type sequence again, all the way to the end. So that's an inversion. An insertion is just that. It's the addition of a piece of DNA from somewhere else. Again, you compare the mutant sequence with the wild type sequence. You get down to the GGA. Here's a GGA. And then you're looking for CAT, and you see that that's way down here. And what's in between those are 6 additional nucleotides that came from somewhere else. They have no sequence similarity to the wild type. So that's an insertion of DNA that doesn't normally belong there. So is that clear? And I'm spending time on this because many of you did not get the right answer to this question on the quiz. And this type of question will probably come up again in future quizzes or tests. Any question or confusion about how you spot a mutant sequence? Okay, several questions asked about the components of macromolecules. What are the 4 components of macromolecules? What are they? Anybody want to volunteer what the 4 main components are found in macromolecules?
Patrick Dowell here.
Uh-huh.
Carbon, hydrogen, oxygen, nitrogen.
Right. The acronym is CHON, C-H-O-N. Yeah, those are the main components of macromolecules. Now, some of you may say, well, there's also in some of them sulfur and others have phosphorus, but those are not the ones that are there in the majority of macromolecules. Carbohydrates, for example, have CH and O, but not N. Anyway, that is the answer to that question. If you're thinking about what are the elements in macromolecules, it's CHON. All right. Now we'll move from more general considerations to the 3 topics that are covered here, starting with membranes. And one of the questions that came up repeatedly was, what does amphipathic mean? Well, the definition of amphipathic is very simple. It's a linear molecule that's hydrophobic on one end and hydrophilic on the other. In the process of outlining the information in this section of the course, we encountered amphipathic molecules 3 times. I'd like to remind you what those examples are. A phospholipid bilayer, bile acid from the gallbladder, and soap. So let's just look at each of those and see why each one is amphipathic and what the functions are, because the functions are very different. In the case of a phospholipid bilayer, the phospholipid you see here on the left is a long chain hydrocarbon tail, which is nonpolar, and a polar head, which involves a phosphate group and another group. So the polar head is compatible with water on both sides of the membrane. The hydrocarbon tails are incompatible with water, and that's what makes it into a membrane, separating the interior of the cell from the exterior, both of which are aqueous. You have a watery cytoplasm, a watery extracellular environment, and the membrane has this amazing ability to separate those two because the phospholipids are amphipathic. When we talked about digestion, ran into this again. Now you remember we have, in the food we eat, we have all the macromolecules. We have carbohydrates, proteins, fats primarily. The carbohydrates and proteins are not a problem for digestive enzymes because they're all soluble in water. And the enzymes that digest them operate in an aqueous environment. Fats, however, are not soluble in water. They're not accessible to digestive enzymes. That's why we have bile acids. Here you see an example of a bile acid, glycocholic acid. It is a steroid on one end, nonpolar. It's an amino acid on the other end, highly polar. That makes it amphipathic. So how does it function? Well, when fat is in the diet, it needs to be solubilized. It needs to be made compatible with water. So this amphipathic molecule makes an emulsion of the lipids in the food you eat, and that emulsified material can be solubilized so that the fat molecules are mixed in water, and enzymes then, which are water soluble, can digest them. So is that clear? Anybody unclear how digestion of fats requires an amphipathic molecule? Okay, then soap. We talked about soap, how when you wash your hands, an amazing thing happens. Soap molecules are amphipathic. They have a long chain, a nonpolar tail, and a hydrophilic head. So if you wash your hands and you have dirt on your hands, which is oil-based, and most dirt is, a micellar form, which means that the soap molecules orient with their hydrophobic tails in the nonpolar substance you're trying to wash off, and the hydrophilic heads are sticking out into the water that you're using to wash. So once the micelle lifts the dirt off your skin, the water washes it away. The soaps are also called surfactants because they break the surface tension of water. Okay, so amphipathic is clear to everybody. 3 different examples of of amphipathic molecules and the function of each. It's always important when you know the structure to also know the function. There were a number of questions about membrane transport. Does active transport require a membrane? And I think so. I don't know how it would happen without a membrane. But the questions revolved around what can affect the transport of materials across a membrane, and what is the role of cholesterol in the fluidity of the membrane. And for that, I point you to this slide, details of the fluid mosaic model of a membrane. And listed here on the left are 3 factors that influence the fluidity of the membrane, and the fluidity determines how well things pass across it. So a main factor is temperature. As temperature rises and falls, fluidity rises and falls, and the speed of the movement of objects across the membrane rises and falls with temperature. The degree of unsaturation of the fatty acid tails in the phospholipid bilayer, the degree of unsaturation also promotes fluidity or affects permittivity. If the molecules are completely saturated, they're long, straight, and stiff, they're rigid. And there's not much fluidity there. If there's unsaturation, 1 or more double bonds, the tails are kinked. They bend, and when they bend, they force spaces between adjacent, between them and adjacent tails. And the spaces will allow for more fluidity, more flexibility in the membrane. And remember, the membrane is not a solid and it's not a liquid, it's somewhere in between. And it has to have that sort of dual nature in order to be semi-permeable. Okay, so that's how the degree of unsaturation promotes fluidity. What about cholesterol? Well, cholesterol can cut both ways. It's like a buffer. It's not a buffer, but it acts like one. So at low temperatures, cholesterol increases fluidity. So as the temperature goes down, fluidity decreases. Well, cholesterol mediates against that. And at high temperatures, fluidity will increase because the temperature is rising, the membrane becomes more watery. Cholesterol mediates against that. It decreases the fluidity at high temperature. And it does that at high temperature. It raises the melting point of the phospholipid membrane so that the membrane stays together, it doesn't melt, because cholesterol raises the point at which it can melt. And the opposite, at low temperature, the cholesterol makes spaces. So in the same way that the bends in the tails make spaces, cholesterol, you see the little yellow objects here in the membrane, cholesterol can make spaces to keep the membrane flexible at low temperature. So those are the factors in response to the questions that you posed. Those are the factors that affect transport due to the effects they have on fluidity. Membrane has to have a degree of fluidity, of ability to flow in order for things to be transported. If fluidity is too high, membrane falls apart, the cell dies. If it's too low, it loses permeability altogether. Even though it maintains its structure, nothing can go through it at a low temperature.
I have a question. So does that mean that it cannot be too high or too low? It have to be in the middle, right?
Yes,
Okay.
yeah, that's the point here. That membranes are essential, and transport across membranes is essential for life. And so if the temperature, or if the fluidity, because of these factors, if it's too high, the cell is going to die. And if it's too low, the cell is going to die. So it has to be between point A and point B in order for the cell to be alive. Okay, anything else? There were a number of questions about what types of molecules can cross a membrane and what are the mechanisms by which they do it. So for that, I refer you to this slide on the movement of small molecules. They move by 3 different processes: diffusion, passive transport, and active transport. And you see that illustrated by the different colored dots. So the red ones illustrate diffusion. Diffusion is passive, and it's usually very slow. And of course, the higher the temperature, the more fluid the membrane, the faster diffusion would be. But diffusion is always from high concentration to low. You see lots of red dots outside, just one inside. So it's from high to low. Passive transport is the same thing. It does not require energy, and it goes down the concentration gradient from high concentration to low concentration, but it does it through a channel, through some structure that's a component of the plasma membrane. Now, in contrast to those two types of transport is active transport. And the contrast is striking. First of all, energy is required. For the first two, it's not. Active transport requires the expenditure of energy. And notice that now the movement is against the concentration gradient, where you have a high concentration here of blue balls outside the cell, low concentration inside, and yet this transporter is moving them against the concentration gradient. It's moving them from low concentration to high, and it's hydrolyzing ATP to release energy in order to do that. And I've got a quote here from somewhere I found it, that gases such as oxygen and carbon dioxide can pass freely through a membrane, and water can pass, but very slowly, and water passes by the process of osmosis. Remember, osmosis is nothing but diffusion. It's diffusion of water, but through a semi-permeable membrane, and that's what the plasma membrane is. It's a semi-permeable membrane. The larger the molecule, the slower it can pass through the membrane. Then you have a worksheet on membranes, and on that worksheet are some problems, some questions about tonicities. So let's talk about tonicities. They are hypotonic, hypertonic, and isotonic. Isotonic is easy to understand, and these tonicities are always with respect to water. If the concentration of water inside the cell is the same as outside, the cell is very happy, and it's called isotonic. If the water inside the cell, the concentration is low, that means the solutes in the cell are relatively high, and water flows into the cell, and it puts the cell under stress. It's turgid, and in fact, the cell could burst under hypotonic conditions. Hypertonic is the opposite. The water concentration in the cell is high. The solute concentration is relatively low, and water flows out of the cell, so the cell shrinks. It's called plasmolysis. And you can see these phenomena with either red blood cells, shown here under hypertonic conditions. The red blood cells shrink. Under hypotonic conditions, they swell up, and in fact, if the osmotic pressure is too high, they will burst. Same thing is illustrated here to the right, except that in addition, plant cells are shown, and they can't shrink because of their cell wall, but the cytoplasm inside the cell can shrink under hypertonic conditions, where water flows out of the cell. Any questions? One question asked during transport, can the cell membrane be damaged? I think the act of transport alone does not damage the membrane, but other things can. And how is transport affected if there is a problem with the cell membrane? I'll point to the CFTR protein as an example of something that will happen if there is a problem. So if the CFTR protein, if the gene for it is mutated, so the protein does not function properly, then there's a problem in the membrane. And what is the problem? Chloride ions don't get transported outside the cell properly. The mucus outside the cell cannot be diluted with water, becomes very thick and sticky, and that causes problems all over the body. And there were a couple of questions about electrochemical gradients. Here is a statement of one of them. How does it form? And is it because of differing concentrations of ions on either side? And in this case, the questioner answered the question. It is. And I refer you to this slide taken from one of the lectures. We're looking specifically at sodium ions with a high concentration on one side of the membrane and a low concentration on the other. So that's a chemical gradient based on the concentration of solutes, high on one side, low on the other. It's a chemical gradient. But because sodium ions are charged, the fact that there's a high concentration of them on the inside of the membrane means that that membrane now takes on a negative charge. And the opposite side of the membrane takes on a positive charge. So the difference in charge is called the electrical gradient. And between the two, it's called an electrochemical gradient. Okay, I guess no questions about that. Another question, what determines the type of endocytosis that it is? Is it pinocytosis, receptor mediated, or phagocytosis? Well, you have a ready reference point with receptor mediated endocytosis by recalling coronavirus, the spike protein binding to the ACE2 receptor. If there's an ACE2 receptor on the plasma membrane that the spike protein can bind to, that binding is a signal for the membrane to invaginate and keep going and finally pinch off and form a vesicle. And now the virus is inside the cell. So that's receptor mediated. That's an example of it. I'll talk more about that later. Now, in the case of pinocytosis, it's fluid uptake. It's not just water. The fluid has something in it, like protein molecules or fat molecules. Anyway, they're dissolved or suspended in water, and the content of the water can be a signal to the membrane to do the same thing, to invaginate, pinch off, make a vesicle, and now the molecules that were outside are inside, and they can be transported to wherever they're needed inside. A phagocytosis is reserved for large objects, like bacterial cells. Again, the cell itself, the bacterial cell, is a signal to the membrane to invaginate, which it does, pinches off, makes a vesicle, and brings the bacterial cell inside, where the vesicle can then release it, and the bacterial cell can cause disease if it happens to be pathogenic. So those are the three types of endocytosis. I'm going to jump over the next three on membranes, and we'll come back to those if we have time. But in fear of running out of time, I'm going to switch now to the most important questions that you raised about cystic fibrosis. First of all, what causes it? That question was asked several times. And you know, what does cause cystic fibrosis?
A mutation.
Genes.
Okay, Kaylin, was that you? Answered. And someone else said something. What was that?
Patrick, genes, recessive traits, genes, stuff like that.
Well, what about the genes? What about the gene?
Seventh chromosome.
Yeah, that's where the action happens.
It has to do with the protein or the amino acid placement, or what's going on with the protein.
Yeah, that's for sure. But the protein is one step downstream. What happens before the protein?
A mutation from each parent passed on.
Yes, that's the critical thing to remember about cystic fibrosis. It is a genetic disease. It only happens if there's a mutation. And it only happens if that mutation is in a particular gene, the CFTR gene. So cystic fibrosis is not contagious. It does not spread around. It's not vectored by anything. It's just a mutation. And you mentioned heritability. There were a number of questions about how cystic fibrosis inherited. I have a little diagram here to illustrate that. If you take two parents, a father and a mother, and each is so-called heterozygous, that is, each on one of their chromosome 7s is wild type, the other has a mutated CFTR gene. The other parent is the same. One of its chromosome 7s is normal, the other has a mutated gene. Now, due to random assortment in meiosis, what you end up with is 25% of their children will be unaffected. That is, some children will get 2 wild type genes. 50% will get 1 wild type and 1 mutant. And the remaining 25% will get 2 mutant CFTR genes. These are the ones that will show symptoms. And there's a 25% chance that any child of 2 such parents will have symptoms of cystic fibrosis. Now, if only one of the parent is a carrier, one of the parents is a carrier, what would happen?
You don't have a
Is
50
that half?
chance.
Would any of the children have cystic fibrosis?
What was the question?
If only one of the parents was a carrier and the other one had 2 wild type genes, what would happen in the progeny?
The child wouldn't have cystic fibrosis.
There'd be no symptoms,
It
but
could still be a carrier.
Oh,
Yeah.
he can still be a carrier. No.
right, there could be carriers in the progeny, but they would not have symptoms of the disease. Okay, so that's,
Is it possible to like skip generations, or is it always going to be like that, where there's going to be one child that's either a carrier or symptomatic, or is it possible to like have it pop up in like grandchildren?
no, it doesn't matter which generation you're talking about. The distribution is always the same. For any child, there has to be 2 parents. And if both of those parents are carriers, then what you see here on the screen are the probabilities that will always happen. So if you say these children, now take them to the next generation, if you have a wild type mating with a double mutant, then you can pass the disease on. In fact, all of the progeny would be either carriers or have symptoms. Yeah, so it doesn't matter. And we'll get to fertility issues in a little bit in cystic fibrosis, but the fact that males who have cystic fibrosis are infertile means that they're not going to pass their recessive genes on.
Professor
Yeah.
Bennett Taylor, what if the two parents were just carriers? Would that mean that the offspring would automatically be a carrier too?
Well, that's what you see in this diagram. Both parents are carriers.
But I mean, if they weren't showing symptoms.
Yeah, carriers don't show symptoms.
Okay.
So carriers don't know that they're carriers unless they have their DNA analyzed. They would only find that out before the days of DNA sequencing. They would only find that out if they had a cystic fibrosis child. All right, to change the subject somewhat, some questions about the path of the CFTR protein. For that, go to the slide, which you see here, it's in your slide set, which shows the path from DNA to membrane and protein and membrane. So the CFTR protein is encoded on chromosome 7. That DNA is transcribed and translated into the CFTR protein in the ribosomes and gets processed there, is transported then to the Golgi apparatus, where it is decorated with sugars, long polymers of sugars, which are necessary for it to go further. It gets transported through a number of vesicles and finally ends up embedded in the plasma membrane. Now, each of you is going to have a project where you'll have a mutant, and each of your mutants is going to be blocked or affected at a different stage of this convoluted pathway. Some will make no protein at all. Some will make a protein that aborts before it gets to the plasma membrane. In other cases, the protein is made, but it's inefficient or unable to transport chloride ions. So with a reduced or absence of chloride transport, you get the symptoms of cystic fibrosis. Okay, so if you, on your own time, you can look at this picture and assimilate what it means. What are zymogens? Anyone?
Katrina Bibbard, aren't they inactive enzymes?
Yes. They're a form of an enzyme that is not active. So a question arose, can cystic fibrosis affect production of zymogens? So before we talk about that, of the digestive enzymes, which ones are produced as zymogens by the pancreas?
Can you repeat the question?
Of the pancreas produces a number of different digestive enzymes to digest the food that you've eaten. Of these enzymes, which ones are produced by the pancreas as zymogens? Think of the types of substrates that these digestive enzymes have. Carbohydrates, proteins, fats,
Is it protein?
proteases. The proteases are produced as zymogens because, why? Well, it's because the pancreas is made up largely of proteins. So if it produced proteases, it would digest itself. Therefore, it produces proteases as zymogens and releases them into the duodenum, the small intestine, where they are activated by other enzymes so that they can now start digesting proteins. The other digestive enzymes do not have substrates in the pancreas, so they can be produced as active enzymes and exported into the small intestine where they digest food. A couple of questions about pancreatitis. That is an inflammation of the pancreas. Can it lead to diabetes? Well, there's been a certain amount of work studying that question, and the evidence so far suggests that, yeah, it can. So what causes diabetes? It's a lack of insulin, and inflammation of the pancreas can reduce insulin and could lead to diabetes. There was one question, why is pancreatic cancer so deadly? Because it has a reputation for being almost a death sentence if you have pancreatic cancer, if you're diagnosed with it. And the reason is that pancreatic cancer has no symptoms until after it metastasizes. And very often the symptoms are not from the pancreas, but from some other organism that has become cancerous because the pancreas is cancerous. Someone asked, is it possible to live without a pancreas? And the answer is yes. In fact, it's apparently not too difficult, but you do have to change your lifestyle considerably. And by that, I mean all the functions that the pancreas provides, you have to get somewhere else, primarily in the food you eat. So you need to have enzyme supplements. You have to have hormone supplements to take the place of what the pancreas would do. And quite a few questions about how can you prevent or cure cystic fibrosis, since it is just a mutation. Can you fix that mutation and correct the disease? And the answer is yes. Especially now with the availability of CRISPR, it's fairly easy in a test tube to take cells that are mutant and clip out their mutant CF gene and replace it with a wild type. So that part is relatively easy and it's been done. The hard part is to take what you've done in a test tube and deliver it to a living organism. Well, one way to do that is to edit the fetus. But that is considered unethical, and I don't know of anyone aside from a lab in China who has ever done that with a human fetus. So what are alternatives? Well, you can engineer wild type cells in a test tube and then try to get those cells into a living human. You could do that by putting them in nanodrops of oil, much like the vaccines that we're going to talk about soon. You can try to deliver them through a virus vector, but in some way, the corrected cells have to be added to a living organism. And that's a very complicated and sort of a probabilistic. You try as many things as you can think of and hope something works. Several people ask about symptoms other than on digestion. We talked a lot about digestion in class, and you're going to run into this question when you research your project. But yeah, there are a lot of symptoms, some of which I've listed here. The lung is affected in a major way, causing breathing problems. As I said earlier, males are infertile because they don't develop a vas deferens to transmit sperm. Often patients have osteoporosis, and that's due to the fact that their bones cannot metabolize properly because of an ion imbalance. And what is the ion? It's chloride. And usually patients have a very salty sweat, again, because of ion imbalances. Let's see, so we got here.
Got a small question, Professor.
Yeah,
So we've been looking at cystic fibrosis and the pancreas a lot. How does it get to the lungs and why does it affect the lungs? I was reading that it causes a lot of chronic bronchitis and stuff. Why?
it's the mucus.
Ah,
Remember why mucus forms, and that mucus is everywhere on the cell surfaces, and mucus is fine as long as it's fairly dilute so it can flow. But if there is an impaired transfer of chloride ions, then there's less water on the surface of cells, and the mucus gets very thick and sticky. And if you can imagine, in lungs, you have all these tiny little airways, little air bags, air sacs. If they get all clogged up with mucus, then air exchange becomes real difficult. Can cystic fibrosis be treated? Again, you're going to research this on your project. There's a company here in San Diego called Vertex, so you should look on its website. It has a number of chemical treatments for cystic fibrosis patients, which do not treat the mutation, they treat the symptoms.
Okay. I had a question.
Yeah.
It's Justin Hansen. So you said it has been done in China though. Do you know, does anyone know like if it was like successful or what even happened with that?
I don't know. It wasn't done on cystic fibrosis. It was just done
No, just done.
Human fetuses.
Huh.
And I can't remember, but it was so.
Did they get in trouble or something for that?
Yeah, international conduct.
Yeah.
There, as far as I know, there have been no other reports from China about that.
Okay.
Now, people do it with monkeys, and that's not considered unethical. All right. Yeah, we're getting short on time, so
Professor, I have a question.
Yeah.
So it cannot be treated. This is Anna Mendez.
Uh-huh.
My question is so it can't be treated at all, not even if you're not fully, you don't have, like, let's just say the person is not fully mutate, like doesn't have that full gene and they only have a little part of it. Can they still be treated? Or can they still be cured if they have only part of it?
I don't know what you mean by part of it. If they have the mutation, they're going to have symptoms. The symptoms can be treated. And when you look at, for example, if you look at Vertex's website, you can see that they have a number of chemicals that do treat the symptoms of cystic fibrosis. So they don't correct the mutation. The disease can progress, but it can be treated. And but that only works on certain mutations. And again, when you research your project, you'll find out if there are chemicals available to treat the symptoms of your mutant.
Oh, okay, thank you.
Okay, let's go quickly now to coronavirus. A good question. What's the difference between SARS-CoV-2 and COVID-19? SARS-CoV-2 is the name of the virus. It's one of many different severe respiratory viruses that are known for humans. And it just happens to be the most virulent one that's ever been found. COVID-19 is the name of the disease. So if one is sick from having contracted SARS-CoV-2, the name of the illness is COVID-19. Where did it come from? Not known with certainty, but the best evidence says that it did come from China. It probably came from the city of Wuhan, and it was probably transmitted to humans from an animal. The animal could have been a bat, could have been a cat or some wild cat relative. Nobody knows for sure. There were a lot of questions about vaccines. So I'd like to spend the rest of the time addressing vaccines. Are vaccines effective against new variants? As you know, there are a number of variants that are more contagious, and all the evidence so far says that the vaccines that are currently available are effective against these new variants. Will a new variant arise that will be immune to the currently available vaccines? And the answer is, it's just a matter of time. The reason for that is copying errors. Copying errors occur at some constant frequency. And we have all these coronaviruses copying themselves in all of these human hosts, billions of copies. And what that does is create a huge reservoir of mutants that are potentially immune to the vaccines. So that's why there's such so much pressure now for people to get vaccinated to reduce the number of copies of the virus. The fewer copies there are, the lower the chances of a variant that will be immune. Where are the new COVID strains coming from? Well, I've just said so. They're coming from mutations, and the mutations occur because of copying errors. There has been some reporting in the media that the Johnson & Johnson vaccine is less effective than the others. That's not really true, because there are so many variables here that you can't compare the different data sets directly. One thing that can be said with some certainty is that the J&J vaccine is equal in effectiveness for those who have serious infection, serious symptoms. But the advice is that you take that all of the vaccines are effective. For example, the J&J vaccine on its worst day is more efficacious than the best flu vaccine. And people get flu vaccines every year and they're happy about it.
So
Uh,
Professor, with that, if you get the coronavirus vaccine, do you need to still get the flu shot? Since you said that one's like better, do you think that covers you for a little while, or is it still a good idea to get the flu shot?
They're two completely different things. Yeah, so if you should get a flu shot regardless, you should get a coronavirus vaccine regardless. You need
Well,
to,
yeah, I know the coronavirus vaccine, but I didn't know if the coronavirus vaccine worked against like flu
No,
too. If it was
no, it does
since it's
not.
better, it knocks out. Okay.
Are there long-term side effects and will booster shots be needed? Don't know. This disease has only been known for a year. And so that's too short a time to know if there are long-term side effects. So far, booster shots have not been needed, but who knows when that might turn up that they are.
Professor,
Yeah.
I have a question. Forgot to mention on this vaccine part. Can the vaccine be given to someone who's ill with COVID, and would it benefit them to get the vaccine if they had COVID?
As far as I know, the answer is yes.
Okay.
But I really don't know any more about it than the public in general. It's just whatever I read in various journals.
I had a question, Professor. My name's Abby Beck. If you had previously had COVID and then you got the shot, do you think that that would affect you differently than someone who hadn't had COVID previously and also got the shot?
Yeah, it could. I don't know that there's enough evidence on that to really draw a conclusion, but yeah, having had the disease, you will have some antibodies, which is the same thing that a vaccine does. It induces antibodies. So they should be complementary. In other words, the antibodies you already have should be just in addition to the ones that are induced by the vaccine. Can vaccination cause death? No, I don't think there's any case of vaccine causing death. Why is injection into a muscle? Well, think about that. What are the alternatives? If you didn't inject into a muscle, where would you put it?
Intravenously?
You could do that, but what happens in blood is not conducive to the way a vaccine works. And I'll point that out in just a minute. But yes, it could be injected directly. But if you're going to do that, it would probably be better to inject antibodies directly into the blood.
Professor Taylor.
Uh-huh.
Back a couple questions though. Have they figured out how long the vaccination is going to last yet? Last time we discussed it, you said it was kind of up in the air.
Well, still is, because the time is too short. We've only known the disease for a year, only had the vaccines for a few months. And so we just don't know how long the immunity lasts. It could be a long time, it could be short, but we won't know that until after the passage of time. Okay, I'm going to skip the next couple because we're running out of time. And I want to talk about how the different vaccines work. Now, I went over this in class, but it's a little bit complicated. And so I'll go over it again, and hopefully it will be clear to all of you. So first, let's consider how the messenger RNA vaccines work. Now, I should emphasize that this is a new technology. These are the first vaccines that have ever been based on RNA. All vaccines in the past have been based on protein. So remember, if you look at a coronavirus particle here, its genome is RNA. And one little segment of that genome encodes the spike protein. The spike protein is what sticks out at these red things sticking out from the periphery of the particle. this little piece of DNA encodes that protein. Now, to make a vaccine, what the pharmaceutical companies have done is take that piece of RNA alone, directly, straightforward, package it in a little oil droplet, and that's what gets injected. That is the vaccine, just RNA in an oil droplet. In a muscle cell, that particle fuses with the plasma membrane and releases the mRNA directly into the cytoplasm. And now what you have here is what I showed you earlier. You have all the components of protein synthesis machinery. You have the mRNA, you have the ribosomes, and you have the tRNAs. And so they combine to make spike protein. Well, these spike protein molecules make their way to the cell surface, where, if you go to the next picture, the vaccinated cell now has spike protein on its surface, and that induces the immune system, the B cells and the T cells, to make antibodies. And these antibodies are soluble, and they can travel throughout the body, secreted antibodies. And so now, if this vaccinated person encounters the coronavirus, what happens is there are all these antibodies that the person has which bind to the spike protein. So all the spike proteins are blocked by antibody molecules. Now, if that particle wants to cause disease, up here in the upper right, this is a picture you've seen before. Spike protein, in order to get into the cell, has to bind to this receptor, ACE2, and then it gets taken into the cell. Now, if the spike protein is bound up with antibody molecules, it can't bind to ACE2. That's what the red X is for. It says no binding occurs, no infection. So that's why these mRNA vaccines are so effective. Now, I mentioned that Pfizer and Moderna make mRNA vaccines. They're both about equally efficacious. The problem with them is that they require 2 doses to get full immunity. And because the packaging is in these tiny little oil droplets, they're very fragile. That's why they have to be stored at an ultra cold temperatures, minus 80 centigrade. And most places don't have freezers that cold. So that's a problem. Now, there's another type of vector called adenovirus. This is a much older, tried and true technology. It works for a lot of different vaccines. It's very good at vectoring almost anything that you want into a cell, and that's because adenovirus is highly evolved to invade cells. So what happens in the case of adenovirus, well, let me say first that Johnson & Johnson makes a vaccine based on adenovirus. It has the advantage that it only needs refrigerator storage, so 4 degrees Fahrenheit, and it only requires 1 dose for full immunity, and that is a big advantage. So how does a viral vector vaccine work? Start again with the virus particle. Again, this little piece of RNA encodes a spike protein, and you'd like to get that into an adenovirus vector. Well, adenovirus is a DNA virus, and you can't put RNA into a DNA vector. So what do? You use an enzyme called reverse transcriptase, which converts RNA, single-stranded RNA, to the corresponding double-stranded DNA. So you take the spike protein RNA, make it into a double-stranded DNA, and now you can splice it into an adenovirus vector which is very good at invading cells. And now it releases the vesicles, release the virus into the cytoplasm, but it can't function there. So what does it do? It injects the DNA into the nucleus. With mRNA vaccines, the nucleus is not involved at all, but it's essential for adenovirus. Now, once the DNA is in the nucleus, it's transcribed to RNA, and now we're back to having essentially an mRNA vaccine. The mRNA in the cytoplasm is translated into spike protein, which appears on the surface of the cell, and we're right back to here where we were with the mRNA. So it's basically the difference is in the packaging and how you get the vaccine, the viral genome into the cell. Somebody asked if you get the vaccine, you're still susceptible to the virus. That is not true. The vaccines all work. Does vaccination protect other people that you're around? Yes, it does. If your viral load is reduced because you have antibodies against the virus, then you're not spreading it around to the people that you contact.
Professor?
Yeah.
So that means that so if we have the vaccine, you're saying that I cannot get COVID now?
You're 95% chance you won't. It's not completely without fail.
Okay.
That's what's called 95% efficacy, means that out of 100 people, 100 people exposed to the virus, 95 of them would not get sick, but 5 would. So there's always a chance, although a small one, there's always a chance that the vaccine would not protect you.
Professor, I have a quick question. So if you have the symptom, would you still getting the vaccine, or it's not working anymore?
If you have symptoms, yes, you should get the vaccine. If you have symptoms, it means you're making antibodies naturally, but you should still get the vaccine. And if you're symptomless, you need the vaccine. If you're carrying the virus without symptoms, you still need the vaccine. I have run overtime. There are a lot more questions. See if there's anything that's really critical here. No, I've covered the most important points that I wanted to cover. I've put this compilation of questions and answers into Canvas. So if you want to consult this document, you can. So anything else? I have kept you late, but if there are any questions, I can address them before we go.
Yeah.
Thank
I've
you.
got one.
Okay.
So my question is, when you get vaccinated, and remember how the second dose kind of knocked you on your butt?
Uh-huh.
When it comes time to get the next vaccine, if it becomes yearly and we still have to get 2 shots, will we have built up the antibodies so there's less chance of you getting sick on the second dose again?
Well, it's my understanding that the bad reaction that one can get, which I got, is not really a reaction to the virus. It's a reaction to the adjuvant that Moderna puts in their vaccine. The adjuvant is there to stimulate the immune system so that you get more antibodies sooner. But apparently some people can react to that, which I did.
So is it a steroid? That
Adjuvant, I don't know what the chemical nature of the adjuvant is.
all right, thank you.